Hello and welcome to another episode of Momento Malum today. For many crops, there's extensive infield conservation effort for many cultivars and land races of annual crops. There are, of course, many problems with this process, the most common being the same cultivars having multiple names, misidentified pedigree, and a lack of information of the crop's genetic variability. It is no different in sorghum. The different genetic marker technologies have been made to discriminate between cultivars and populations, as well as discover the genetic diversity of these cultivars. This project was taken on by Dr. G as part of a plan to analyze the genetic diversity of sorghum. This project would involve the genetic analysis of 25 genetic lines of sorghum, 5 lines from the 5 main land races. These will be seen on the next slide. These lines will be analyzed using the SSR marker technology and the Gen Survey software program, and here are the results. The experiment found that there is a high level of inbreeding due to the large number of fixed traits. Despite this, there's still a high level of genetic diversity within the land races. Approximately 53% of the genetic diversity stems from the variation within a population. 14% of the genetic variation stems from the differences between populations. The bicolor land race was found to be the land race with the greatest level of genetic diversity. The land race that had the least amount of genetic variation was the Kaffir land race. On the regional level, the Eastern African region has the highest level of diversity between and within populations. In contrast, the lines from Asia have a much lower genetic diversity within populations, but higher differentiation between populations. This is plausibly because the populations are derived from multiple small introductions of sorghum to Asia. Overall, there's too much genetic variation to form distinct genetic lineages within populations. However, there's an exception to this rule. 3 populations from the Kaffir land race, known as K1, K3, and K4, were found to form a genetically distinct group. The results of this project were compared against previous studies. One of these studies was done in 1989 using enzyme markers, a precursor to genetic markers. The results of this older study found a genetic diversity 30 times lower than this study. While it seems bad at first, this was a normal result when comparing enzyme markers to genetic markers across all grass species. This is likely because the genetics of enzyme making are prone to homogenization, while other traits are more variable in nature. This fact was discovered in 1995, and it's generally why enzyme genes are no longer used extensively in the study of genetic diversity. The greater genetic variation found in the bicolor land race corresponds with the physiology studies on these ancient land races. These studies found that this bicolor land race is the most primitive of the land races, and the populations are closely related to the wild ancestor in comparison to the Arab populations. This land race is also the most widespread of the land races, allowing for greater genetic variation to occur within populations, due to the sheer volume of specimens. The Kaffir land race seems to be comparatively more recent, and it's only found in South Africa. These factors will of course limit the gene pool of this land race. The researcher concludes that given the limited resources of the different gene banks, populations from the bicolor land race of Eastern African origin should be prioritized for conservation, since this is where the majority of the genetic variation can be found. And that about covers everything. Thank you for watching. If you want to donate to my channel, the links are below. If you want to buy my book, you can also find that link in the description box below.